Okay. Well, we have a lot of questions that we took from the community on Facebook, so I have all of those here, and I'm sure we have many in the audience, and then we're monitoring them online. So if anyone online has questions, please also share those, and we'll ask them if we have time. So to get started, thank you all for your great talks today. I think it's incredible how far we've come over the last few years. I remember when Stephanie stood up as a student in Dr. Weaver's lab presenting on ketone esters, and now you're the Angelman syndrome lead for a gene therapy program. So we've all been here together and kind of grown together, which is pretty awesome. All of us have really come a long way in a short period of time. I'm going to start with Wendy. You are our keynote, so I'm going to let you take the lead. A question for you. As parents, and all that you heard today and all that you know that's being developed in Angelman syndrome, we are luckily put in front of an embarrassment of riches potentially, which a lot of rare diseases don't have, and we appreciate that more than anyone knows. And making a choice for your child and the questions our families have for if all of these programs are out there, and by the time phase 3s start and new phase 1s are coming, choices are going to have to be made where if you choose one, you can maybe never see another, and if you choose another, you may have to wait for approvals before you can see something else. And it's really hard as an advocacy group, certainly, to guide families, which we never do. And they're also asked to talk to their local doctors who understand very little about what's happening in Angelman syndrome, certainly, so they also can guide very little. And as a clinician who's not working in Angelman syndrome from a therapeutic perspective, you're the only one I think on the stage who can actually guide parents and answer this question. What would you say is the best place for them to turn to ask the questions on what I should choose for my child? Because those that are running the trial may not be running all the trials and are going to be biased in their choices as well. How do you answer that to your patients that would be asking you that question when they're a newborn screen positive and the parent says to you, which one should I choose? Don't tell us which one you would choose, but what is the advice you would give to that family? Can we put the mics on, please?
It's a very good question, and as I was listening to all of them this morning, that was actually what was going through my head exactly. So I don't know in terms of who the trusted voice is specifically. I'm not saying it should be me, but I agree with you, Allison, is that it should be someone who's unbiased yet knowledgeable, and that's a tough one to do. So I think as a community, you'll probably end up providing guidance, and within this, I think of both the upside and the downside, and I think about things that are one-time versus permanent in terms of this, and I realize that there are some things that'll be a bridge to a permanent solution, and every family has to make their own decision based on what risks they're willing to take and what they're looking for in terms of a permanence or durability for that. So we've heard everything from things with pills, with ASOs, to gene therapy, and with doses. I don't think we know yet. It's kind of the Goldilocks situation, not too much, not too little in terms of risk and benefit. And I always say that I'm really impressed with the brave families who are willing to be first in to do these, because we don't know how it's going to go. You've heard this over and over again. But I think everyone's going to be looking to and should be thinking about what risk they're willing to tolerate. And in terms of doing this, there are certain things where you do it once and that's your shot. And so you've got to think about whether or not you should or want to do that. And for some people, that one and done is a good thing. And so they very much value and weight that. But for others, it's sort of nerve-wracking to do it and to get one shot on goal. And so I think you've got to really think carefully in terms of doing that. I will say, just watching for other rare diseases, it's been interesting to watch everyone play chicken. And so what I mean by that is that for other conditions where we're not sure about the initial doses, I have been watching with hesitancy who's going to be first in to do that, sort of everyone waiting to let someone else be the brave first person to put in. And I've seen trials where everyone's still waiting. And so I don't know how those are going to go at the end of the day. But there is going to have to be someone who's brave enough to move forward and do that. And we're going to learn from each other as we're doing it. So, but I hope there are I'll call them Sherpas, but people out there who I do think are quite knowledgeable in terms of the field and the therapies, and I hope a few of them will be able to be unbiased and guide the community with that. It would be good for you guys to invest in a couple of those people to guide you.
Yeah, and that's exactly right. So I think as advocacy groups, we're going to come together with boards of people around the table that can talk and learn and understand, to Jennifer's point, consent and what is known about each product, and really reading the details about what is in that animal data that parents need to know and need to understand in order to be able to make those very, very clear decisions. And so hopefully we'll be able to have at least enough handouts to explain what each one is in a way that's understandable and digestible, and then the doctors running those trials can really dive through some of those details with each family. But it's going to be tough, and we get this question all the time. Okay. The next question I have is actually somewhat related, just kind of going back globally, and then I'll go into the details of each person. But this is for Dr. Wilson. So as part of the Orphan Disease Center, not as part of the gene therapy program, but your work with so many rare diseases, some of which you have gene therapy programs for and some of which you don't yet, or are guiding on what's the right thing to do for them. You have probably touched an equal number of rare diseases as Wendy. And you made a choice to jump into Angelman syndrome in 2016. And you see a lot of monogenetic disorders. What encouraged you to take on Angelman? Because it was a decision that took a lot of thinking about, being that you don't take on every disorder that people approach you about. And where are you at in your mind about that decision that you made? We're thankful you did take it on, by the way. But.
Can't say no to you.
No one can.
No, I think, a couple things. One is there's a pretty good animal model. Some diseases have no animal models. But what fascinated me, and which we're exploring as well, is that the nature of the genetic lesion is such that there are strategies other than just treating a loss of function and targeting the antisense in different ways just expanded the potential therapeutic approaches to basically cover almost any modality. I know of no other disease that has that kind of optionality that Angelman has, and that was a big part of it. What's also very important when we engage with communities is that the physician scientists who've served the foundation of this community are good people. And now you may say, well, what do you mean by that? There are situations where some of the leading academicians are not very nice people. And that really creates a huge problem. And this is a nice community, that the physician scientists that form the foundation of this cooperate, work together, and then finally the frontier is really to have a foundation that can provide leadership. And I can tell you that there aren't many places like diseases that has a FAST. And it just makes it so much easier because there's so much churn and drama around a community in which the physicians are fighting with each other, and there isn't really a leading alliance-building foundation. So disappointed that we haven't been able to make more progress, but have gotten to the point where we're comfortable with moving forward, and we try to be careful about that. And the work we've done so far, most of the work is ahead of us. But we need the community of patient advocacy groups and physicians to make sure that happens.
Thank you. That's a really great answer. You can say that again, the last part, if you'd like. This is a question I'm going to direct. Maybe I have this for two of you, but maybe I'll start with Becky Crean. So as more patients will be needed in phase 3 clinical trials, of course, so we start with small studies, and it's going to grow to need potentially 100 plus patients in trials, and more and more enrollment is going to be necessary. Will you allow patients that were in other ASO trials to enroll in these phase 3 trials if the other ASO trials ultimately don't get to phase 3? if that makes sense. So if another trial, for the audience, this is a question asked online, if all 3 ASOs, if they don't all get to phase 3, but someone was in one that didn't get to phase 3, will there be an opportunity for them to enroll in the studies that do get to phase 3 so they don't lose their opportunity for having been in a different trial?
Okay, so we're talking about different drugs.
Different drugs. Let's just say one of the 3, ABC, does not get to phase 3, but A and B do. Would a child that was in phase 3 be accepted into one of the other 2 company trials?
Right.
Because now they're no longer going to have drug if that program was canceled.
You're going to hate this answer, but it depends. We heard that a lot yesterday, didn't we? It's going to be very dependent on each individual program, each drug. So what we're learning from these early ones are really about safety, tolerability, dose levels, dose intervals that really kind of build on and inform the next phases. And as we sit here today, because some folks have talked about data, but nothing's published and have been scrutinized and really gone through the rigor of looking at this data, we don't know much about all of these compounds. I mean, we know more internally about our own, much less about the other ones. And so we have to make decisions as we're progressing our programs on what we can tolerate from the outside coming into them, because these are all still clinical trials. They're not treatment. We're still learning each step along the way with each trial. And so we have to be as strategic and concise and thoughtful about that each stage of the way. The goal of all of these is to understand to the best of our ability what our compound is doing. So it'll get tricky as we get further along. We get more information, which gives us more confidence that the compound can tolerate more outside influences coming in, but we're not there yet. So each company will have to make a decision on that. And part of it will be based on timing. If something's been published and we know more about a particular compound, whether it's an ASO or some other compound that an individual with Angelman syndrome might be taking at the time, those decisions will have to be made at that time. It could be a timing thing, it could be a safety thing, it could be something about another compound that wouldn't interact well with your current compound. So really in these clinical trials, they're not treatment, they're clinical trials, we have to be thoughtful about how we gather information about our drug, and so we know the most that we can about our drug and how that'll help people. So I'm sorry, it will depend though.
Well, I think that's a great answer, actually, because I think what's important from the community side to advocate is that we beg anybody, particularly for ASOs, because there may be a changeover of one doesn't make it and one does, or 2 do and one, or whatever the combination. If any program, God forbid, I hope they all make it, doesn't, we beg you to share at least half-life data that you have so that the other companies that may go forward can make decisions for the patients that now will not receive therapy on whether or not they understand that washout period. So enough information is at least shared on failed programs, any failed program. Share as much information as possible so that the children aren't left behind and the companies that are going forward can make those educated decisions, because a lot of things that failed don't ever get published. And then we're left with not knowing, so then the answer is no, because we don't know. Do you know?
Well, I will say that with the growing robustness of clinicaltrials.gov, it's been a beast and an archaic system for a long time. They do require you to publish results in that system. So whether you decide as a company to publish them in a peer-reviewed journal, it doesn't matter to clinicaltrials.gov. If you're going to run an interventional trial and you are required to list it on these websites, and I know that it's similar for ex-US, there is a certain period of time once that study completes that you have to post results. And it's information such as that. It depends on the question that the trial is trying to ask and what your endpoints are, but especially in these earlier ones, those are the major questions. So that data does get disclosed publicly, whether we choose to publish in a journal or not.
That's great, because I think that will give some comfort to the community to know that educated decisions can be made long term. That was a question we had from a couple of families. Next question is, I'll ask this to Brenda, if you would. All the phase 1/2s are open label, obviously, and we talked about that today. Do you intend your phase 3, or do you know if your phase 3, if you get there, which we all hope you do, would also have a placebo arm?
Thank you, Alison. It is an important question. I think we heard this morning about the drug development process. Usually, yes, phase 3 are randomized controlled trial. This is required also from the health authorities. We need to show that our drug is better than placebo. So even if now is premature to comment on, especially for rugonersen, but in general, this will be a requirement, yes.
Okay, great. I figured that would be your answer. Next question here is, if patients are enrolling in a phase 1-2, and I'm asking this only to the ASO folks because, or actually you can also answer this, from Neuron's perspective for repeat dosing, for one and done it's not going to be, I know the answer. If patients are enrolling in phase 1-2 as they're making these decisions, which are hard decisions to do, and then a phase 3 chooses a couple of the best doses that were evaluated in phase 1-2, and now they're in, the phase 1-2 kids or adults are in open label, will you increase the dose of those individuals or decrease the dose to what you determine the best dose for the phase 3, or do they need to remain on, if they were in the first or second cohorts, the lowest potential doses for phase 1-2? Will you adjust them based off of the new information you have on your phase 3? So maybe I can ask that to you, Emil?
Well, we're already doing that. So the patients that start in our lowest dose cohorts do titrate individually up to the higher doses until they get to a certain level of efficacy. It's part of the way we're exploring the dose. So it would not be appropriate to have people, just because they got randomized to a low dose level, to keep them there where you've already determined that's not an adequate dosing. Most people would get adjusted. So that's already happening, in fact, in our program because of the way it was designed. But if any design we would have, we wouldn't deny people the proper dose once it's determined. I would say one thing about dosing too, though, Allison, is that we always act like there's a dose, but truthfully there's usually, should be think of it as a range of dosing. And I actually think there's going to be some level of individual titration, which is tricky, hard to do in phase 3, but at some point in the program I think people may need individual titration too. So I'd add that as another variable.
This is a question for gene therapy. So Stephanie, I'm going to ask you this question if you don't mind, and I think Jim, you can probably answer this as well. If a patient, we're going to go the opposite direction. If ASOs get approved before the gene therapy does, which at this rate might happen, and they're now on an approved ASO, would they qualify to be in a clinical trial for a gene therapy because it would, once an ASO is approved?
I think that's a great question, and our CMO is here if he disagrees with that answer. But at this point, I can't say we can say anything definitively, but we definitely are taking into consideration as standard of care will be changing. We know that we're going to have to look at the changing environment, that it's a consideration. I can't say yes or no right now. I don't know if Lee wants to share anything additionally, but we know that that's probably something that's coming. I don't know if there's anything else I can help address, sorry.
I mean, I think SMA really showed us where they, went. Some kids are now getting both, but once they were both approved, when one's in a trial and one is approved, that's where it gets pretty complicated. Do you want to answer that too, Jim?
Just one comment as it relates to the SMA emerging story about 2 approved therapies, one in ASO and gene therapy. I think where there's going to be pushback are the payers. They're going to want to pay for one or the other, not for both. That's what I'm hearing.
Which might be why, if they're getting one that is approved, they may want to be in the clinical trial for the other one because then it's free to the family or the payer. Okay, that's great. Thank you. And the next is about placebo for gene therapy. And I'm going to ask this to Jennifer and to you, Stephanie, because they're very different types of gene therapy potentials. We hear a lot about open label for phase 1-2 and placebo control for phase 3. Is that similar for AAV gene therapies, or are they leaning more toward with gene therapies doing placebo in the phase 1-2 as well? So maybe I'll start with AAV, and then Jennifer, you can answer that for HSC.
Yeah, so it's a really great and important question, and what we found is that there doesn't seem to be uniformity globally with all of the health authorities. And recognizing, obviously, this is a global indication, we're a global company, we want this to be a global program, we're definitely continuing to have, and will continue to have conversations with the health authorities to ensure we can bring the best global study design forward. We recognize that would be potentially a burden, for sure, for families. And I just can't answer that today. It depends on where we can start, but it's a big, important question for sure.
Jennifer?
Thanks. I can speak to the two approved lentiviral HSC gene therapies that I mentioned, Skysona and Zynteglo. They were both open label studies because there was concern about putting a patient through a transplant that was really just a placebo. So there, where they're taking the cells out of your body, putting the gene therapy in, and then transplanting them back, they did not put patients through a sham or a control.
That's important to know as well. That would be a hard thing to sham, I imagine. Okay, this is for Jim Wilson. As you've personally experienced setbacks in drug development, and I think you have a unique skill set in the sense that you're in academia doing a lot of the basic science and then also translating them, starting companies, giving them to companies and seeing them go through the process of IND enabling and trials, which most early phase preclinical programs in academia don't really experience. So you've experienced the setbacks of gene therapy through your career, and companies dropping programs, taking programs, companies being in the clinic and then being dropped for financial tumultuous times that we're going through right now. And so sometimes things are hard to scientifically advance. What advice would you give FAST in our community to ensure that we always remain the priority for programs that are looking good scientifically, but unfortunately may not always be a priority for other companies?
Yeah, especially today, ask this question. It's a really important question for many, many rare disease programs where because of economic conditions, market conditions, decisions made by management, they can't raise money to continue existing programs. If we rely on biopharm, which we do, to ultimately commercialize products, we as foundations or the academy lose control, or they go slower than you want them to go. I think a couple things. One has to do with to make sure the academic labs that work with companies that there is clear diligence requirements, which are, it's almost an oxymoron. Diligence and clarity doesn't exist, and I know companies don't like that, or to apply pressure when a company drops a program, that the rights go back out of the company so that the university can find another sponsor for them. I think that's really important. The other is, what not only FAST, but many other organizations are doing. We're working with Friedreich's ataxia and CDKL5 deficiency, where the foundation or the parents said, well, we're going to start a company, and rather than donate, we're going to invest in the company, and we're going to work with academics like you did with genetics. And others are doing that. They're following your lead, and the notion, and they're doing it not to get a return on investment. They're doing it to make sure that we stay focused on the task at hand and progress the program to the point where it would be almost impossible not to find a biopharm to take it on. And I would encourage FAST to continue to, especially when you get into the translational mode, to, if you do have the resources to do that, and you can put together the collaboration in order to progress it to first in human, where you have more than safety readout. But others are doing that. It's a pretty amazing sea change.
Yeah, I've been asked to give some talks about this, and the titles they always want it to be is that patients take the lead. And that's a new concept, really, and it's wonderful to see so many organizations that are doing this. So I'm glad that you agree with that approach. I'm going to ask this question to the paternal activation folks, so I'm sorry to go back to you guys again, but this is a big question for our community that I've already asked you guys before, but I'm going to ask you again. I think I ask you every time I talk to you. Is 6 to 7% of our population has not qualified to receive any therapy based off of their rare genotype of UPD or ICD, and particularly for paternal activation, of course. And we all know the theoretical risk of overexpression of UBE3A with 2 paternal alleles. What would you advise for us to do as a community to, one, mitigate that risk for you, if we had to do it to encourage you to treat those individuals, and ensure that all genotypes will have access to your potential therapeutic throughout the course of your drug development program? Is there something we can do that hasn't been done, other than time in humans before you make that decision, so that we can ensure every genotype is appreciated in your clinical trial? So I'll start with you, Emil.
Well, we've been considering this carefully, and I actually don't think there's anything the patient community needs to do. Like, I think you're thinking something experimental to de-risk, and we feel like the data we've seen in the non-human primate, in my mind, kind of de-risks this, and that's because in non-human primate, they have one maternal copy that is working, and when we unlock the paternal copy and both are expressed, what we see is the maternal expression goes down. If you look at the mRNA concentration, so you see equivalent levels of maternal and paternal. And my belief is when we're releasing, or let's say enabling expression, we're not forcing the expression, and that whatever other regulatory elements exist are regulating UB3 production. Therefore, we think that that will probably protect the UPD patient from having just going full bore 2x expression based on that, because that's exactly the situation. You have 1 active, you make 2 active. If you start with 2 inactive, I think it gets you to the same place, 2 active chromosomes in 1 cell. So, and remember, most of the body, both chromosomes are active. It's really only in the neural cells that this is imprinted. So we're not concerned about it. We just feel like we need to start with a simple story of the severe patient to demonstrate what we're doing, and then we'll look at those other populations in separate studies and in separate kind of cohorts of studies, so that to get to the answer. But right now, I would say nothing. I think we are at this time comfortable that we would be okay.
Do you want to answer that as well?
Sure.
Becky?
I'll take a little bit different perspective. I think I don't disagree that this isn't the burden on the community necessarily. I think, as we heard yesterday, and there's more research that needs to be done, maybe in the lab and some of the animal lines or cell lines, and that can better inform on some of those decisions. That is the risk, this theoretical risk of overexpression. And as we sit here today, we have these thoughts about it, and we can make some assumptions based on what we know about those that have 1 paternal copy. But we need more data to guide that. And so I think there can and there is more research going on. I think the second thing, though, is, and the community is already doing this, they're participating in some of these CSF trials. I mean, the way we're going to know most about what UBE3A expression is in the CSF right now, what with the ASO trials. And so the community's participated in that. They've completed 2 of those, which is phenomenal. Never been done before. We haven't measured it before. And it's given us a way to develop the tool that now we can use, the assay tool that we can use in the clinical trials. So as we start to understand not only just levels in the system and how they may change over time, now we can look at them in response to a treatment. And when we know more about how they respond to a treatment, those with the 1 paternal copy, then what we can do is try to use that data and information to potentially, maybe we get to a point where we're dose adjusting, we're targeting a certain protein level, and we can use that tool to adjust our dosing so that even those with 2 paternal copies can be included, and there's not a risk of overexpression. So I think we'll have to triangulate this. I think it's being done. I think the community's helping and being involved in that. And the next step then is understanding it from a treatment approach.
Great, thank you. I'm sure we'll have more UPD-ICD questions because it's very, very carefully considered in this community, of course. This question is for Dr. Jones. When does Neuron plan to open clinical trials outside of Australia?
Good question. So I did forget to mention, our phase 2 trial is actually quite small. It's an N of 20. So we aren't planning to open any international sites for that study. That will be completely in Australia. But then all going well with the phase 2 and we continue on to bigger trials, then it's our expectation that we'll be in other geographical regions. And as I did note, we do have an IND open with the FDA for Angelman syndrome. So.
Great, so for phase 3 potentially is where we would see it?
Yes, correct.
Okay.
Yes.
Good, I'm glad your study is only 3 months. Okay, and then one more for you. Can you explain to the community, there's an over-the-counter compound that people talk about called Brain Shield, NZ Brain Shield, which is a blackcurrant supplement that people say has some of the same derivatives that are in, or are the mechanism of action of your compound. Can you just explain that how they're not the same and why people should or shouldn't be going and buying Brain Shield?
Yeah, so just if people aren't familiar with it, I'll explain what that is. So NZ Brain Shield is a particular brand of these types of supplements, dietary supplements, so they're not approved treatments. And they come in different brand names as well that you'll look up, and they have, but they have a substance called anthomycin, which is derived from plants, derived from black currants, and that has naturally occurring cyclic glycine proline. It's actually from the plant. It's just like plants can create lots of other kind of elements that might be useful as pharmaceutical elements. So that is just something that naturally occurs from that substance. But it is, again, these are supplements, so again, they vary in terms of how much of that is in each different type of supplement. What NNZ-2591 is actually a synthetic version of cyclic glycine proline. And again, as I said, it's the exact same molecular structure, but then also has that slight change, which makes it more useful develop as a medication. So from our perspective, the difference is that we are sort of carrying this forward as a medication under a regulatory framework and sort of demonstrating each of those steps to show that it actually, one, does the same thing on the preclinical studies as naturally occurring CGP, and that you'll actually be able to demonstrate that it is safe and effective. So the other things are their food supplements. So again, how much is in it or whether it's good for you or not, it hasn't been demonstrated in a clinical trial. But it is a naturally sort of, its nature, and it just happens to occur in these types of plants.
Great, thank you. Why don't we open it up at the table? I have a whole lot more questions here, but why don't we're getting close on time, and I want to make sure we have some audience questions. Tom's hand is very high, right over here. This light is a lot.
Hi. An ASO question, if I might. With multiple ASO trials now taking place, and on the surface all appear to be doing the same thing, what are the key differences we should be aware of when weighing up what trial to potentially enter? And are those differences the reason why, for example, Ionis are able to look at deletion and mutation, and Ultragenyx can't?
The question is there a difference between the ASOs that leads us to try different targets? I think our approach in general would be, and this is what genetics team's decision was, that the deletion type is the most severe type and it's the most consistent type for which to figure out what's going on with efficacy, whereas the missense other types can be a lot more variable in what the phenotype is. So it was about getting a consistent phenotype and figuring out the drug dosing and then expanding. I don't think that's the difference between the ASOs. I think that's just a choice one can make and how well you want to control the population. I think there are programs where you could go broad and learn in the first phase 2 study, but I think in this case, the focus on deletion point gives us a little more ability to figure out what's real and what's not when there isn't a lot of progression on their own in the severe type for natural history purposes. There are differences in the ASOs. Our ASO targets a different area, different chemistry. The choice we've made was based on Scott Dindo's data, which suggests that closer to the 5' end, the message is a little more potent in the knockdown. And we think that that's true, and that's the basis of the targeting. But I don't have a lot of knowledge about the other ASOs in terms of chemistries, but I know that there are differences. But most of the clinical decisions are really about the process of determining whether a drug works or not in an open label setting, trying to develop at least some consistent sense of response in a more limited population before expanding.
My question is for Brenda. Given the point of allogabat, how it's looking at other deletions across, is there thinking about combining allogabat with, say, the ASO to see what the effects would be together?
Thank you. It's a great question. And in summary, the answer is yes. Like that's definitely something for deletion genotype, right? Like if you look at the mechanism of action of allogabat, and you're very familiar now with the antisense, with rugonersen, there's a potential clearly of combining, like we need to show that both are efficacious and have a good safety profile, but that's definitely an option that we might want to consider for deletion genotype. Clearly we know that for deletion, like as I explained earlier, it's not just the UB3A, the problem. There's clearly there are other genes, some we know, some like there's still ongoing research, we heard like these days, but to answer your question, there's definitely that potential, yes, which is very exciting.
I'll follow that up with, will they be excluded or included to be in your clinical trial if they're in the Roche ASO study as well? Are you looking at it totally separately, or are you looking at a dual
No,
compound
yet, right?
study?
We cannot look at them together at the moment. This will come. The first thing, as I said, we need to show that rugonersen is efficacious and with a acceptable safety profile. We need a phase 3. The same thing we have to do with allogabat. And then we can think about combination.
Over here. Thank you all for the great talks. This really is for anyone working on any of the nucleotide-based drugs, but it was triggered by Dr. Crean's talk. I'm really curious scientifically, when you're doing lead selection and winnowing down to a candidate, what are the types of dose-limiting off-target toxicities you see with these modalities, and how different are bad actors versus the eventually selected molecule? Because they're so different than when I think about small molecule selection.
Yeah, and chemistry is beyond my area, so I'll try to talk through this. So it's actually quite remarkable the way they do this, and I don't know all the details about it, but in doing this for 30 years, we've developed through trials and tribulations a pretty good screening system to screen ASOs. And it starts with a very broad net to look at ASOs that first target what you want it to target. Forget about safety, efficacy, all of that. Like, does it get to the place we want it to go to? And what you learn along each of those steps is some of them don't do what you want them to, so you throw them out. Some of them you see safety signals in them. You see tolerability issues. You see off-targets. We do a lot of off-target studies to look at if there's anything else that it's targeting in the system that we don't want it to. So it's a very systematic approach, and I'm not describing it eloquently at all by any means. The funnel is the best I can do with it, but it's a process that we've refined over many, many years and looked at ASOs in many different indications, and that helps us, we take those learnings forward and helps us each stage along the way to select out ones that only do what we want them to do, don't do anything else, and give us the best profile.
Hi, maybe one for Brenda and Rebecca. It's great to see the progress that's being made in the trials, and as you look to be thoughtful in data disclosures, do you think that we might be able to see, should the expectation be for part 1 of the trials to see data before or after completion of the part 2 elements? Thank you.
So the question was, in understanding how you're waiting for the trials to complete before you share data, are you going to be sharing data at the end of the phase 1/2, or are you going to be essentially waiting for the end of the phase 3?
So I can,
I think, but part one, I think he means the phase 1/2 versus the phase 3, right?
No, parts.
I don't understand what he's saying either then. Parts of what?
If there's different components of it.
So
For the-
I
you're asking about
can
ESOs.
answer it both ways, I think. I think both of our trials have a part 1 and a part 2. Part 1 is the short, kind of 3 months or less dosing, and then you roll into a long-term extension, which we're calling part 2. And so we kind of think of it as all one trial because it's gathering long-term safety data and efficacy data on the drug. So it's likely that once we complete both parts of that trial, then we would disclose the data. Obviously, we're evaluating it internally and making decisions about the next stage. So the data is being used, but when we publish it would probably be once we complete the entire trial. For the very same reasons we wouldn't disclose data during an ongoing trial, whether it's in the beginning or middle or end. We want to wait till the completion of it to really preserve the integrity of that data that we collect throughout the life of that trial. And so, so then if you're asking about a phase 1 versus a phase 2, once that trial is completed, our process has been to publish that data once we've looked through all of it, and we'll publish that. So that could be while another phase is ongoing. There could be a gap in between, and it just depends on the type of program that we're running. But once a particular study has completed out, that our practice would be to publish that data.
yeah, I agree with Becky. I just want to remind the community, right, that the objectives of a phase 1, like this phase 1 or 1/2, whatever they're called, is safety and tolerability. That's the first objective. So what I explained today is that for rugonersen, like we are pleased to say that the safety profile is acceptable. So in one word, I basically already answered that question, even if I didn't show you the data. Right? That's what we are assessing with this study. Of course, we look at we call it exploratory. It's an open label. There is no placebo. We look at efficacy as well. We have scales, we have questionnaires, and but this will be shared once the study is completed.
Hello. Oh, good afternoon. First, I'd just like to thank all the speakers for sharing their data today. The progress is evident and exciting, so I just wish you all the successes possible as you continue your endeavors. My question is for Dr. Wilson, and it sort of piggybacks on Allison's earlier question about UBE3A overexpression. In your work, is overexpression of UBE3A protein in the gene replacement a concern? And if it is a concern, what sort of steps are you taking to try to precisely control the level of protein in the cells in the therapy?
The overexpression is always a concern when we are unable to completely recapitulate the normal regulation of the gene. But we will be looking, and we have been looking very carefully at the toxicity of overexpression in toxicity studies, including non-human primates. And if there's direct toxicity to the cell, you're going to lose expression pretty quickly. And that'll be at least an indirect measure of that. But there are other measures of neurotoxicity that we're pretty careful to evaluate. If we see that, but we haven't seen it yet, including in our primate experiments, then one would need to find a way to modulate the expression. And we do encounter projects where overexpression is a problem. And then we go into a second phase of development, and that is weaker promoters. So you tone down the level of expression. We call it the crappy promoter project, because generally you try to generate too much. But we haven't seen the need to do that so far with the fairly extensive studies that we've done so far.
I have a question for Jennifer Panagoulias regarding the stem cells. Could you clarify if, when you modify the cells, if it's only adding a new UB3 gene for the deletion patients, or whether you would replace additional genes just like GABA, SRPN, or any other genes basically with a same maternal allele? And if you don't, is there a projection to do so in the future? Over.
I can only comment on the work that's been done by Dr. Anderson's lab thus far, which was only to replace the UB3A gene, not any additional genes. And since we're still in an early phase, we don't have any plans to try to expand that.
Okay. This question is for Becky. You keep mentioning the CSF and UB3A in the CSF. Have you seen, can you share with us any data that you have about the concentrations that you see?
Oh, yeah. We actually presented on that. I guess that was on, yeah, it was not in this forum, was it? I think Rob has presented that, I may be misremembering, but either Biogen or Ionis has presented some of that data, maybe this summer at the ASF, but we have made it public, and we're happy to provide that. And I don't know if Allison, if you're sharing any of that.
I'm not sharing anything from
ABOM. Yeah.
I know the answer to the question, but
yeah,
I don't want to share
I guess the short answer is it is measurable in the CSF of patients with Angelman syndrome. We looked at both deletion and mutation so far, and we are able to measure it in the CSF. The assay is pretty sensitive, so we can measure pretty low levels. We've got some optimization to do on it to make sure we can capture really, really low levels in case those exist. It's a small sample right now. We've got more work to do on it, but it's really promising. It's going to be a really helpful tool for clinical trials.
So if I can comment on that, I think that what we are missing is age-matched controls. That's what we don't have, right? So we have values for individuals with Angelman syndrome, but we are missing normotypical CSF protein, right? We don't have like that key number for individuals of the same age. That's something that will definitely help.
Hi. Before everything, I just wanted to say thank you all. This has been amazing. This is our first conference, and just hearing everything that you have to say has been amazing and immensely helpful for us. But I did have a question, and I don't know if it's exactly for this panel, but it's regarding the clinical trials themselves. We have encountered that it's been a little bit difficult to understand the clinical trials page and the places where these trials are being held, and how the process is done. I know that you're focusing on the science, and we're super excited that you are, but is there any, as this moves forward and they're gonna open more spots and things like that, is there anything that the companies are doing to make sure that this process is a little bit better to understand, of who is being picked for the trials, how is it being done? There's make sure that there's obviously a transparency on who's being picked, all that. And then I had another question regarding the medical trials that are happening outside the US and in the US. For those who may be willing to travel, is that an option for people, let's say, that will be willing to travel every 3 months to, let's say, Italy? Is that an option, or are there rules? I'm sure there are rules about this, but just wondering. Thank you.
I imagine that's going to be company specific, so we can get started. Brenda,
yeah,
you can start.
I can start there are multiple questions. In general, drug development is very complex. Clinical trials are very complex. You can definitely look at the clinicaltrial.gov page, I think you mentioned. It's not super easy to understand, but you have apart from your patient organization, you have your doctor, so your Angelman syndrome specialist. Just sit down with your doctor and ask the question. They're there to answer this question. Like you need to really understand. I'm looking there, but I don't know. I couldn't see exactly where the question was coming from. Okay. So yeah. So like you really have to go to your doctor, like, and take advantage of, like, sit down with your doctor and discuss, ask all the questions that you have, because they are there for you. The second part was about the move, like, eventually moving country to country.
Cross borders.
So we discourage that. We cannot stop families. There are issues like with we are administering scales. These are translated and validated in that particular language. So sometimes it's really complicated, like it's actually not possible. In other cases, like time of COVID, because like we don't really want like families with an Angelman child to just move from country to one country to another country, or like to state. Ultimately, it's the family's choice. But we, as Roche, like for the nurse, we discourage that.
In the way we conduct trials, we identify clinical sites that have experience with Angelman and potential intrathecal treatment, and they really particularly have to be able to do the intrathecal treatment part and all the assessments associated with that. We set up the clinical sites. They control within their site where the patients are coming from, and many sites won't necessarily take patients from anywhere. Our Canadian sites, for example, didn't want to take any US patients. One did, some would, not all. So for the redosing, we did have some, but others wouldn't. They would only do Canadian patients. Since many of the sites have their own patients already, it's very unlikely they're going to want to take patients that they don't know when they have patients that they do know at the site itself. So it's really, we pick sites, and the sites end up deciding on patients that qualify for the program. And currently we're in UK and Canada because those were territories where we could get approval to proceed. In the US, we're working at a different dose level and working on opening the US. But our expectation in phase 2 is to start adding some other countries to distribute in Europe and South America to gain experience and build a network of sites that would be involved in our phase 3 ultimately. But I guess the concern I heard in the question was, is it fair to get how to get access? Is it like an inside track or something? And it's really only, we pick clinical sites that have experience and they end up picking the patients. And as a company, we don't tell them, we give them criteria, but we don't tell them who. We're not controlling their choice. It's really up to them. And I would say generally they don't really want to have people moving, but I will also say in some of our other programs that are less common, like MPS 7, we had to fly people from Portugal, Brazil, to the US to conduct the trial, to find enough patients. So we do move patients around when the PIs do not have enough patients on their own to make it work.
Great, thank you. Well, we're out of time, so I'm going to end it here, and please feel free to come and engage. They're going to be here all evening, I hope, and at the gala as well, so don't be shy and come and ask questions. But I'm going to end with one final question for each of you to answer in 1 minute or less. Every year I do, and hopefully you're prepared. One word. No, one sentence. So if you can answer, as a departing statement, our AS community is obviously ready, willing, and able, and wants to join and be a part of this mission, and they all want to see. If people are sitting in this room, they generally are very interested in transformative treatments for their children and their loved ones. And they want to support research, and they want nothing more than to see a transformative, life-changing therapy, and many have a high-risk tolerance to do that. As they are waiting anxiously, what is one statement you would make to them in terms of what a departing comment for them to hold the optimism to keep going until they have their chance, which may or may not work, but ultimately are waiting? Stephanie, do you want to start, and everyone can just make one final remark that the community just can hear to wait to speak to you again next year?
So be involved. And it seems like you guys are doing that by being here, but share your story, educate yourself, come to the conferences like you guys are doing now. And then and be part of patient registries, natural history studies, and ultimately clinical trials. And that will help us bring these novel therapies to your families.
It's amazing how things have changed, but what is helpful here, among other things, is competition, and you got a lot of competition here.
I remember you told me competition breeds competition. Yes. All shots on goal, don't stop trying every option. You said that about 5 years ago. Here you go.
I would say, be happy, something's coming.
Don't worry, be happy.
I didn't say that. No.
I share the same, and I would say also like be involved and stay objective. That's is very close to my heart. We need to be happy because there are so many trials in Angelman syndrome that, as you said multiple times, years ago we wouldn't have even dreamed of, right? And so now we have this opportunity, but we need to stay objective.
I would just say, there's a lot of different roles to play in this, and it's not just being in a clinical trial. There's all sorts, by being here, you're involved. And so I would just say thank you for that, and being involved in a way that works for you and your family and that you feel comfortable with. But no matter what it is, you're making a difference. Everything you're doing has made a meaningful impact in the treatment of Angelman, and I think this stage can attest to that.
Well, I would say my own family is affected by Angelman syndrome, and I have never been more excited about the opportunities that exist right now. 4 clinical trials now, 5 next year, we heard. So keep pushing.
As an outsider, you don't know how lucky you are that you have so many advocates, such great leadership, and so many groups here working. So there's a lot of hope that you should have in the room. I'm really hopeful.
And I'd just like to echo with, that there are many ways to contribute, and small things matter. They really do. Someone asking you, like, go out and do a survey, and you think, is that going to really make a difference? They really do, because the things that we've done here is the sort of collective process of all those things having been done before. So if I want to say it in 2 words, it's hope and resilience, because we talked a little about up and downs today, but those are the sister sort of companions, hope and resilience together.
That's great, thank you. So there's 4 things you need to do before you leave today for the gala. 1, a survey outside on economic burden. Number 2, a survey on motor function and what meaning that has for you. Number 3, a survey on quality of life. And number 4, search and rescue, sign up. It takes 1 minute. If we don't know where you are and who you are, they don't know where you are and who you are. And it's very important we know who lives around the world with Angelman. And if you're in this room and you're not documented that you have a child living with Angelman syndrome, that's a shame, because it doesn't document where the need is. And the need for research has to be learned, and it can't be learned unless we know where you are. So 4 things for you to do when you walk out there at the vendors, and then you're done until the gala. Have a good day. Thank you.